My name is Guru. I'm a junior at Somerset County Vocational and Technical High School, and I worked under the mentorship of Dr. Vishnu Amaran Samara. I wrote his last name here. He's a assistant professor at the University of Chicago in pathology, and we worked on the role of EZH2, which is an epigenetic genome in prostate cancer, and the clinical development pipeline for several drugs regarding EZH2. So I'm going to go over what prostate cancer is, what epigenetic regulation is, EZH2 specifically, diagnostics, how scientists are targeting it, and also the clinical pipeline and the stage we're at. So prostate cancer is the second most commonly diagnosed cancer in men worldwide and remains the fifth leading cause of mortality, which is cancer related. Its behavior varies drastically because some people have a very mild form of the disease, while others can mutate into more advanced forms, such as CRPC, which is castration-resistant prostate cancer, mCRPC, metastatic castrate-resistant prostate cancer, and neuroendocrine prostate cancer. And the issue with these advanced forms is we can't treat them with the usual cutting out testosterone or castration that normal prostate cancer, and that's where epigenetics comes in. Essentially, epigenetics is changing gene expression without really changing the DNA sequence itself. So these can include DNA methylation, which you can see on the right side. It's adding a methyl group to DNA. And although it's just like a CH3, people might not realize how much of a change that can make in DNA. Um, it also includes histone modifications, in our case, specifically H3K27ME3, which is a type of gene. And histones are the molecules that the DNA is coiled and wrapped around. And essentially, they regulate whether the DNA is available to transcribe. So, changing when it's available to transcribe and how it's available to transcribe is really important. Chromatin remodeling reshapes nucleosomes and it exposes oncogenic regions. Oncogenic regions are areas where, if the gene is acted upon correctly, it can lead to tumor growth. And together, all of these things are responsible for lineage plasticity, which is essentially what I talked about before, the mutation into different phenotypes, particularly in our case, neuroendocrine prostate cancer, which cannot be treated by targeting hormones around the prostate. So EZH2 and PRC2 were the main topics of our research. PRC2 is uh PRC2 is called the polycomb repressive complex 2, and essentially it acts upon the genes in DNA and basically reveals it so that it can be transcribed into different proteins. EZH2 is, oh shoot, sorry. EZH2 is a section within PRC2, and it's the main reason why it acts here. So it's the aggressive and metastatic. EZH2 has a canonical function, which is the regular function. How it works, it just represses different proliferative states, meaning it like doesn't want cancer to occur. However, when mutations do happen with EZH2, there can be non-canonical functions, which means it does the opposite. It promotes tumor survival by silencing oncogenic repressor or silencing genes, which are the genes that fight back against cancer growth. Over time, this drives lineage plasticity, including a transformation to the neuroendocrine stage. To diagnose prostate cancer at this, like what we usually do is we give a blood test, and this detects the overexpression or underexpression of prostate-specific antigen, which is it's a protein that is found within the prostate gland. But if cancer does occur, tumors cause overexpression usually, and this means it starts leaking into the bloodstream, and then we can see if cancer is there. However, we can't really determine what type of prostate cancer it is, so it doesn't give any genomic data. To solve this issue, there are two types of tests that can be done. So first step is sorry. The first test is IHC, which is immunohistostaining, and it's on the right side here. Basically, we can detect whether EZH2 or H3K27ME3 are upregulated or overexpressed which correlates with the high Gleason score, which is how dangerous the cancer is essentially, and if it will metastasize. We can also use RNA profiling, which is more advanced, and we go through the genetic sequence of RNA, which is how DNA is expressed at the end. And if we see elevated levels of EZH2 expression, then it's pretty obvious that there's some EZH2 correlation with the prostate cancer. Another new area of research is liquid biopsies. It's essentially a blood sample of the prostate region. And because tumors are uncontrolled, they tend to leak out their DNA or ctDNA and also cells themselves. And we can actually do tests on those cells, and that's what a liquid biopsy entails. So now that we identified what the issue was, we can understand how to target it. So there's some direct inhibitors which directly bind, as you can see here, to the EZH2, and it causes downregulation instead of upregulation, which means it silences it in a way. So this molecule is called tasmetostat, and mevrometostat is also very similar to this molecule, and it reduces the gene expression levels. There's also dual EZH1 and 2 inhibitors. So in the same complex, there's two types, but usually we focus on EZH2, but now they're focusing on both, called valmetostat, which you can see here working. There's also PRC2, which is the entire complex, and this can be targeted as well. There's protac degraders, which eliminate the EZH2 protein entirely. So even though it's expressed, we eliminate the proteins after it's expressed. All of these approaches have the same goal. It's to shut down the epigenetic engine, which causes aggressive prostate cancer expression. So now for like the last set of things, the clinical pipeline is essentially how the FDA goes about determining whether we should give the drug to actual patients or not. Right now, we're at phase 1 trials of EZH2 inhibitors like tazemetostat, and they do show safety but limited efficiency alone or monotherapy. However, when we combine them with therapies such as AR inhibitors, which are androgen receptor inhibitors, PARP blockades, and other inhibitors, they've shown stronger ways to like block out prostate cancer. Using this, we can understand like how prostate cancer can be solved in the long term, and hopefully it does lead to a result. So, in conclusion, EZH2 is a central epigenetic driver for aggressive and therapy-resistant prostate cancer. There's still challenges such as redundancy with EZH1, adaptive resistance. So as we end up targeting these cells, our bodies are going to end up evolving to it because we are all living things and cancer is a living organism. And also biomarker variability and potential like toxicity as well inside our body. Future strategies are using liquid biopsies more because they're kind of a like not very well known right now. And also targeting EZH2 has a potential to shift treatment of lethal prostate cancer towards more manageable prostate cancer, which we can just treat through surgery. So yeah, I'm actually writing a paper about this, but I haven't finished the paper in its entirety, so I decided to present a small slideshow. Uh, Dr. Amaran isn't here, but I really enjoyed working with him as well, and it really showed me like an interest towards research. I'm sure someone like mentioned before that they wanted to be a surgeon before. I also want to be a surgeon. I still do, but now I also kind of want to engage in research when I'm in undergrad years or in med school years. Thank you so
Nice, nice, wonderful, wonderful job, Guru. Um, I'm so glad that this internship opened new perspective areas of interest for you. So Dr. Richa Tiwari, he raised a hand. I think she has a question for you.
Yeah, very quick, but Guru, wonderful work. I really enjoyed going through your slides. Loved it. Also, one question, are you aware of any EZH2 inhibitor that's already being used for any sort of patients?
Yeah, basically what I was saying before at the end was we're in like the phase 1 clinical trials. So
No,
they
no,
are
that's for prostate cancer. That's right.
Mhm.
Are you aware of EZH2 inhibitors that are already used for any other
Yeah.
indication?
So I pretty sure they used it for upper body cancer as well. I'm like blanking right now. I forgot the specific cancer,
Yeah,
but
no, no, no worries at all. I mean, I enjoyed it. I thought that you might like to learn more about the use of EZH2 inhibitors in
Mhm.
follicular lymphoma. So it's kind of a blood cancer, and you may be able
Yeah,
to get more perspective from there. But amazing job. I really enjoyed your slides.
thank you so much.